P53 is a protein that is designed to protect the body against cancer mutations, acting as a tumour suppressor. However, the protein itself is often at risk of mutating. If the protein becomes mutated, it can cause serious harm to the body by inactivating the protective function of not only itself, but other tumour suppressors, P63 and P73. In a study, Zhai and colleagues investigated whether the group of mutant proteins are especially harmful to the cell, and whether the P53 mutant undergoes aggregation with P63 and 73 proteins. To complete this study, the researchers stained P53 proteins in a human cell with specific antibodies that carry a fluorescent marker. This allowed them to observe the proteins within the cell. The staining showed normal P53 proteins localizing predominantly in the nucleus of the cell. There is also decreased nuclear distribution with mutated proteins. However, there is a substantial increase in the cytoplasmic localization, which suggests the mutant P53s could not enter the nucleus. The fluorescent staining further indicated that the mutant proteins formed large collections outside the nucleus of the cell. Using this method, it was also found that mutant proteins were structurally destabilized and had an increased natural tendency to aggregate. Due to the mutant P53, it encouraged the accumulation of P63 and 73, preventing them from performing their roles with transcription of the genes involved in cell growth and apoptosis. According to the various authors, the new destabilizing function of mutant P53 proteins is a direct result of their newly increased predisposition to form aggregates with other various tumour-suppressing proteins, as well as other P53 proteins.
There's a link between the aggregation of mutant P53, the lower rate loss of heterogeneity, and the patient survival. Graph A reveals that within 129 cases, the tumours with DNA contact mutations show a higher loss rate of heterozygosity in comparison to those with stabilized mutations. There was also a stronger dominant negative effect on the structurally destabilized aggregating mutations in comparison to the remaining wild-type P53 allele in destabilized germline mutations. Graph B shows the long-term patient survival rate within 623 cases was significantly higher in regards to contact mutations than the destabilizing aggregating mutations. It was also showed that P53 mutations in patients carrying aggregation mutations gained oncogenic function throughout aggregation.
It was found that by using Tango, a computer algorithm used to predict the accumulation sequences of a protein and identify regions with a protein that are susceptible to aggregation. It was also found that mutant protein is capable of inducing co-aggregation and inactivation of P63 and 73 proteins through the dependence of sequence similarity rather than the physical properties of the molecule. This means that the protein depended more on its sequence rather than the actual structure. Furthermore, a study completed by Rajan Bognar and colleagues in 2013 showed that the mutant P53 protein causes co-aggregation with wild-type P53 and repels the tumour-suppressing function of the protein. This is the same effect that mutant P53 has on P63 and 73 proteins.